example, if we are able to start with an undifferentiated human embryonic stem cell and take that cell all the way to a mature neuron, to a mature brain cell, obviously the molecular pathways, the genes that need to be expressed to develop or during human brain development are also recapitulated in that cell culture dish. So if we can understand how normal human cells are formed, we can try to understand how alterations to that development occurs. And the same is true for many diseases. So it's not just hype. We all also hear a lot about stem cell research being hype. It's not hype, because here are just many of the cells that are derived from human embryonic stem cells. Different types of neurons. So these are human motor neurons that could be transplanted into ALS patients, amyotrophic lateral sclerosis, or Lou Gehrig's disease, dopaminergic neurons that could be transplanted into Parkinson's patients, human heart cells, human blood cells. I don't have to convince you that this is blood, right? But so, this is why this isn't hype. We can make human cells that function like their in vivo counterparts that could be transplanted into patients. So where are we today? We understand that human embryonic stem cells have a potential to treat these illnesses because we've seen in animal models improvement of certain functions. For example, we have a rat model of Parkinson's disease when it's transplanted, acute spinal cord injury, a rat model of spinal cord injury when transplanted with specific types of neurons called oligodendrocytes. The rats do recover the ability to walk again. This is documented in video. It has been published. So we know that based on animal models that human embryonic stem cell research offers potential benefit. The first clinical trial is set to happen this year for acute spinal cord injury by a company called Neuron, and it actually went to the FDA. And it filed what's called an IND, an investigational new drug application. And then on May 13th, the FDA put a pause to the study, and it remains to be clarified why the pause is in place. But in other words, the first potential therapy has been applied to the FDA. There are many phase 1/2 clinical trials approved by the FDA for type 1 diabetes, and the leading company here is a California company called NovoCell. Geron's also in California. In adult stem cells, there are hundreds of clinical trials going on for heart failure, bone regeneration, autoimmune disease, many more in preclinical stages, and we are moving forward in other areas using neurostem cells for Batten's disease, which is an extremely rare neurodegenerative disorder, pediatric disorder, and Clive is heading towards the clinics for ALS as well with his work on neurostem cells. We do have some technical hurdles. We need to understand how to produce these cells in large scale, how to differentiate them better. We have to be very careful on the stability of the cell lines. We have to make sure there are no undifferentiated cells in the final product, because in the blank state or the undifferentiated state, like that colony that I showed you, if you inject an undifferentiated human embryonic stem cell into a mouse, you create a tumor, a benign tumor called teratoma, not cancer, a benign tumor. So we need to make sure that when we inject a vial of brain cells derived from human embryonic stem cells into a patient, that therapeutic vial is free of contaminant undifferentiated cells. And there are other quality control and biomanufacturing. Actually, I'd like to thank Derek High for this slide. So here's an example where this woman was blinded. This study is published in the journal Ophthalmology. This woman was blinded by an attack. She was subject to violent crime, had acid thrown, and she was blind. And after stem cell therapy, using adult stem cells, her own stem cells, she recovered her ability to see. And here's an example. Miracle touched me the most because I hadn't seen her at all. here are some cells, and heading towards diabetes, I just want to call your attention for the red dots on this slide, which are, these are beta islet-like cells derived from human embryonic stem cells. And this study was published in Nature Biotechnology, showing that human embryonic stem cell derived beta islets do produce insulin, and they actually produce, so here's the insulin content, they produce insulin. Here are human islets isolated from patients and human embryonic stem cell derived. We're not quite there yet, but the beta islet cells that we can differentiate from human embryonic stem cells behave in a similar manner as their in vivo counterparts. The largest challenge in this study here that's published in Nature Biotechnology is to get these beta islets to do what human beta islets do in our body, which is, you know what? Only secrete insulin in response to glucose changes. So you can't have on all the time. Does that make sense? If a cell is secreting insulin at all times, then it's not emulating, recapitulating what the real beta islet does. The real beta islet only secretes insulin when it detects changes in your blood glucose. But we are on our way. We can demonstrate that we can generate beta islet-like cells that secrete insulin. Beautiful study for type 1 diabetes. Actually, this study is a collaboration between Brazilian scientists and Richard Burt from Chicago, published in the Journal of American Medical Association. Just to point you here, this study had 15 patients, children with type 1 diabetes, 15 patients, 14 patients became insulin-free. So this study, again, you have to be very careful with what you hear about stem cell therapy, right? You hear about trials being executed all over the world. You have to rely on these types of studies, either FDA-supported studies or studies that have been published in extremely reputable journals, which is the case for this. This is the Journal of the American Medical Association. Another study published in Circulation, actually this is a very close friend of mine, I was with him 2 weeks ago in Brazil. This is direct transplantation of adult stem cells for ischemia cardiomyopathies. So a phenomenal study. This study was so effective. It was initially performed, I think, in 20 patients, but the outcomes of the study were so fantastic that now they've enrolled 1,200 patients. So this is the largest clinical trial being executed for adult stem cell therapy. It's being executed in Brazil with a network of 42 hospitals supported by the Ministry of Health, and there's even a registration for this trial at the FDA. So I apologize for the graphic nature of the slide. The other area that we work on is on in vitro systems, hoping that human embryonic stem cells might reveal biomarkers for us to prevent or diagnose birth defects, such as spina bifida. Birth defects can affect 1 in 33 children in the U.S. The birth defects that you're most familiar with are the classes called dysmorphogenesis, which is alterations that are visible upon birth. The ones that we're largely interested in is what we call functional birth defects, especially functional toxicity, when, for example, during development, fetuses are exposed to chemicals in the environment, or the mother takes a drug, and that results in a birth defect. So I'm highly interested in unraveling the causes of autism, better understanding the disease, and hopefully finding diagnostic biomarkers. So here's where we execute these studies. So this is a pure population of human brain precursors. The green here is a stain that only stains brain precursors. So if there were no human brain precursors in this dish, nothing would light up. It's a marker for nestin, a protein called nestin. So these are human brain precursors that we generate. This is from my laboratory. And so we expose these human brain precursors to things like alcohol. And I just got a grant from the National Institute of Health to study the mechanisms of alcohol injury during human brain development. And we also have